So I just want to remind you of the context again. It's always worth coming back to these points of why would we want to look at all of these mouse mutants and phenotype them. And of course, first and foremost, as I've already alluded to, the function of the majority of genes in the mouse and the human genomes is unknown. It's a sobering thought that it's more or less 15 years now since the human genome sequence was published. And yet, there are many, many genes for which we have no idea what they do in either development or biology. And we're remarkably poor by just looking at the genes at predicting the function of what those genes might do. We know that they might be expressed in a particular tissue. But as I said, it's very important that we understand not what genes do in one or two tissues. It's what they do across the whole body system. So understanding pleiotropy, the multiple functions of genes, is absolutely key to understanding biological systems. I've already reminded you that knockouts have been generated and analyzed in only some 30% of mouse genes. There's a huge landscape of gene function out there which we know absolutely nothing about, and it almost seems ironic. It's 10 years since the mouse genome sequence was published, more than 10 years. We still don't know the function of most of the genes in the mouse genome. And where we do know function, I will remind you again, the data for these genes is patchy. It's dependent on the interest and experience of the investigators. So even for genes where we have knowledge about their function, you can go to the database, as you did yesterday. You can go to MGI and ask, is there a phenotype associated with the mutant? It's still patchy information. It's not the broad-based, comprehensive picture that we need to really be able to understand function and to integrate that information with human and clinical genetics. I want to add in another point. This is a much more sophisticated point, and it might be useful for you to go and read this paper, which was in Cell. It's Blair et al. Sorry, I haven't got the actual reference here, but if you quickly write down the title. We're often asked, when you make a mutant in a mouse, you're completely knocking out the function of that gene. So how many diseases in the human population are due to knockouts? Well, not many. Of course, many rare diseases are due to major deleterious mutations in individual coding sequences. But of course, many of the common complex diseases, like the ones that Roger talked about in diabetes, are due to subtle genetic variants in a lot of different genes around the genome. And we're often asked, well, what's the point of making a mutant mouse? It's nothing like a human in terms of the genetic variants that it's carrying. But this paper rather deals with this point because it was a very profound paper, and it showed that there are significant association and comorbidities between Mendelian and complex disease. And this is the key point. Common variants associated with complex disease are in genes which are already known to play a role in Mendelian disease, those individual loci in humans, which when you get a mutation in that loci, you develop a disease, Mendelian diseases. So in fact, there's a very strong relationship between the genes that are associated with complex disease in the human population and mouse knockouts. In other words, if we can find the function of mouse knockouts, that undoubtedly will impact upon our understanding of the variants that are associated with complex disease in the human population. So there's enormous utility in assessing loss of function phenotypes in the mouse. Now, this is a fairly complex paper and complex idea, but I recommend you go and look at this paper because it shows the power of not just looking at subtle mutations in the mouse or worrying about the fact that much of human disease is caused by subtle, common, rare variants in the, common variants in the human population of modest effect. So there is a lot of utility in assessing these loss of function phenotypes in mouse. So what is the IMPC doing? What is its basic premise over the next 10 years? It started in 2011, and the aim is to undertake broad-based phenotyping of 20,000 mutants from the International Knockout Mouse Collection. It's a coordinated activity of mouse centers worldwide, and it's being done in two phases. Phase 1, which we're currently coming towards the end of, was to phenotype up to 5,000 mutant lines in the mouse. And this was seen very much as a development phase where we looked at the phenotyping pipeline, which I'll come on, to look at logistics of how to produce and phenotype the mice, to develop new phenotype technologies, and also to look at the economies of scale that we could generate by doing such a big project. How could we make it cheaper and faster by bringing all of these centers together around the world? And the second phase that we're going into in 2016 is to do the remainder of the genome, to phenotype 15,000 mutants. So to ramp up and do even more from 2016 to 2021. All of the data from IMPC is freely available through a data coordination center, and all of the mice that are manufactured in the IMPC will be available through a global network of mouse repositories. So everybody in this room has free access not only to the data, but to all of the mouse strains that are being generated in IMPC. And as I'll come on to say, that's an enormous international resource that's available to every country in the world, a rich resource of new genetic material and genetic information. So these are the centers around the world who are participating in both the production, the phenotyping, and the informatics. And you can see that they stretch right across the globe, from Australia through Asia into Europe and then to North America. All of these centers are participating in some way in delivering the goals of this project. Now, I'll just come back to this slide that I showed you before. So the IMPC is using embryonic stem cell lines that are generated as part of the International Knockout Mouse Consortium, which has produced today something over 15,000 knockout ES-cell lines. And most of those ES-cell lines carry this very sophisticated but highly of high utility, this allele here, which is called a knockout first conditional ready allele. And it's called that because you can take the TM1A allele, and using Cray excision of the neocassette, you can convert it into a null allele, the TM1B allele, or by using flip, you can convert it into this conditional ready allele, TM1C. Now, this is the allele that we're analyzing in IMPC. It's a fully null allele, and we want to find out what is the phenotype for every gene in the mouse genome when you completely knock out the function of that gene. So it's the TM1B allele. These alleles, in fact, and the construction of these alleles are described in this important paper here by Bill Scarnes and his colleagues in Nature in 2011. And also importantly, and this comes back to a point that we discussed before, all of the mice, all of the mutants are isogenic on one single genetic background. So apart from the mutational change in our gene of interest, all of the rest of the genomes in all of these thousands of mutants that we're making are absolutely genetically identical. And that's obviously very important for making comparisons between gene function and having a solid, standardized genetic base on which we carry out our phenotyping. So this is a very busy slide, but you're going to see it several times. This is a summary of the IMPC phenotyping pipeline that all of these mutants will go through. And there are two parts to this slide, so I'll just focus on the two different parts. There's the embryonic phenotyping pipeline, which I'll come back to later on. And then there's the adult phenotyping pipeline. Now, somebody asked this question before, what do we do in terms of phenotyping these mice? Well, briefly, let me say something about the adult phenotyping pipeline. So we have a variety of in-life tests, which are these tests here, that start at 9 weeks with, for example, open field and grip strength. And they go through all the way through a whole variety of tests in life to 16 weeks, where the terminal tests begin. Body weights are measured all throughout this time course as well. And then at 16 weeks, they go into a variety of terminal tests. Tissues and blood and so on are taken for a whole variety of terminal tests, ending up with histopathology analysis. So there's a huge variety of systems that are actually measured. And for the adult phenotyping pipeline, we in fact put cohorts of 7 males and 7 females through this whole adult phenotyping pipeline. So let's look at some of the other features of the IMPC phenotyping pipeline. We also measure for each mutant that we've created fertility. of the mutant. And we also look at to see whether the homozygote mutant is viable or not. In many cases, when you make the homozygotes for these mutants, they die in utero. There's embryonic lethality. And I'll talk about how, later on, how we phenotype mutants that are embryonic lethal. There's a whole phenotyping pipeline here that I'll come back to in detail. But importantly, irrespective of whether they're viable or not, we look at the expression of the gene, because the nature of the mutant that we make is that there is a LAC-Z reporter molecule within the gene, which allows us to look at the expression of the gene both during embryogenesis. We look at it at 12.5 days into embryogenesis, and also in the adult as well. So we capture embryonic phenotypic expression of the gene, so we know where it's expressed during embryonic development, but we also look at adult expression patterns, so we know which tissues the gene is expressed in and where it's expressed in those tissues. So you can see already we're getting an enormous fund of information about each gene, not only where it's expressed, but the phenotypes that are manifested. Is it embryonic lethal? If it's embryonic lethal, we then do embryonic phenotyping, which I'll discuss later on. So this is a very rich pipeline. IMPC is always trying to make it richer and richer, so we gather more and more information about what happens if you have a loss of functional allele. Now, this just summarizes the different tests in the adult pipeline and sort of splits them up into different areas where we might expect the test to impact upon finding out defects in particular systems, such as neurology, behavior, metabolism, cardiovascular disease, pulmonary, reproduction, sensory, musculoskeletal, immune, and some general tests. So, of course, all of these tests in the pipeline are giving us some information about these different systems and how they're going wrong. And we're constantly looking at this to ask the question, how can we improve it? So we make the mutants, we put them through our phenotyping, we gather the information, and then all of the information from all of the different centers around the world, the so-called mouse clinics, is fed via the internet to the Data Coordination Center for IMPC, which is actually at my own institution at Harwell. So all of the data is coming from all of these disparate production and phenotyping clinics around the world into a central data collection effort where it goes through data validation to make sure that all of the particular aspects of the procedure, the data procedure, as well as the metadata. Remember I talked to you in the first talk about collecting who did the test, what were the environmental conditions, what was the diet, and so on. We have standardized procedures in IMPC, just as we did with Umodic. That's the IMPRESS database. So we make sure that all of the right data is being collected. It also undergoes QC checks as well. There's a lot of quality control done on this data by both automated systems and data wranglers who are reviewing the data. And we get a preliminary set of data, which is released on the internet to the user. But that QC data will also go through a statistical analysis to find all of the phenodiviance for any particular mutant. And then it goes to our core data archive at the EBI at Hingston and Cambridge. And that curated data is also made available to the user on the internet. So there's a whole sophisticated informatics pipeline of collecting the data, checking it, analyzing it before it's distributed to the wider community. And you will have heard some of the aspects of this from Hugh Morgan yesterday. So where have we got to so far in phase one with this project? Well, this slide, as you can see, is a bit out of date. But I put it up not for you to read the numbers here, but rather just to show you that all of the centers around the world are contributing, some more than others, obviously, depending upon the capacity they have, to producing those 5,000 mutants and phenotyping them. But this summarizes the sort of program highlights, and some of these numbers are also out of date. We've probably done about 8,000 microinjections of ES-cell now. We have nearly 4,000 genotype confirmed lines, mutants, which are on the shelf and have been QC'd and are gold standard mutants ready for distribution to the wider community or for phenotyping. A large number of these have been, well, there are more than this now, have been Cre-excised to produce null mutants. But this is the most important statistic, and this is bang up to date as of last week. Nearly 2,000 lines with phenotype information are now available on the IMPC portal. And just to give you an idea of the scale of the effort, this involves getting on for 50,000 specimens, mice and embryos, which have been analyzed, and 750,000 phenotyping procedures have been carried out to date. So there's a lot of data there. But here's another important statistic that I think is important to you and to the rest of the community in terms of getting these lines. So these are mouse mutants that have been delivered to the community. If we look across all of the production centers and the main archives that are disseminating the mutants around the world, you can see that nearly 3,000 IMPC mutants have been ordered to date. If we look at the number of unique genes sent out in the program to date over the last 3 years, this is around about 1,300 mutants. Now that's about 6 or 7% of the total genome in terms of coding sequences. So already you can see there's enormous demand for having a gold standard, well-characterized mutant that's available to the community. So I want to come back to the Umodic program and just recall the fact that, of course, this was what we did in Umodic. We generated mutants, heterozygotes, homozygotes. They went into a phenotyping pipeline that's actually very similar to the current IMPC phenotyping pipeline. And of course, part and parcel of that was we also looked at these reference lines across the consortium to ask how well were our phenotyping procedures working. And the answer, as you will recall, is pretty well. We had a high consistency across the different centers when we analyzed the same mutant. So we've been doing the same thing in IMPC, and although the data analysis is not completely in yet, these are all of the lines where multiple centers have been analyzing different mutants. You can already see that there's good consistency across some of the IMPC reference lines. This task isn't finished yet, but just to remind you that standardization and reproducibility, which is critical in science generally, is also critical to the IMPC, and we're making sure that our standard operating procedures in IMPC work just as well as they did in Umodic. So you'll have already seen this because you worked on it yesterday with Hugh, but you can go, I want to remind you again what you can really go and get from IMPC. Because every day, in fact every hour if you like, if you're a mouse geneticist, we want people to be going to the IMPC website and looking for genes, phenotypes, models of human disease, because there's a rich set of information already in the IMPC, which is almost unparalleled in mouse genetics. So this is the home page. You can search, depending on a gene, you can look for a gene, or you can search for mice that have got certain phenotypes, or you can search for certain models of human diseases. And this just gives me an opportunity to say something about the kind of findings that we're getting here. So of course you can search on a particular gene. You can bring up information about a gene, about its production status, about its phenotype status, and of course you can click on any of these and find out more information. So here's gene LMOD1. Remember this gene that we picked out in Umodic, in fact? Nothing was known about it at all. It had no PubMed hits, and I'm going to come back to this gene later on. But you can already see that if you do a search on LMOD1, it tells you that phenotype data is available, and it summarizes all of the phenotype data down here. I'll come back to this later on, but you can see there's a whole slew of very significant phenotype hits for this gene, which had never been looked at before. I'll come back to that a bit later on to tell you a bit more about it. Of course, you can go and look at what we call a phenotype heat map for each of these genes. You can look and see which is indicated in dark blue, where you get significant hits, in this case in the homozygote, for LMOD1 in the nervous system, adipose tissue, growth, skeleton, and so on. You can also see where there's sexual dimorphism as well, which is an important thing that we're looking at as part of IMPC. You can obviously drill down into any of those parameters. You can use this tool Finoview to look at the data for significant annotations in a number of parameters. Here's LMOD1 again. you can see that it's got a very low startle response when you compare it to the controls. You can see these beehive plots of all the control data, and you can see that LMOD1 have got virtually no startle response whatsoever. In fact, we now know that LMOD1 is completely deaf. It's a deaf mutant. You can look at skeletal abnormalities. You can go and get images for each of these mice. I'm sure Hugh showed you this yesterday. And of course, you can go, I talked to you about how finding out where genes are expressed, both in the embryo and the adult, is a critical part of IMPC. Every gene that we're looking at, we find exquisite expression information. Here's an example of a gene with lovely patterns of expression in the cerebellum. And of course, you can go and look at all of that data and bring it together with all of the other phenotypes that you're seeing in that mouse. So this is the key to mouse genetics in the future. It's not just having bits and pieces of information here and there. It's having all of the information. It's having the phenotype information, what it's doing in open field, what its behavior is, having the images, the radiographs, the expression data, and pulling it all together to understand what a gene is doing, but not just in one gene, in all genes across the whole genome. That's the power of IMPC. Of course, you can look at genes according to phenotypes. You can start from a phenotype. So you might be interested, as Roger is, in diabetes, obesity. So you want to ask, what are all the genes that have been identified in IMPC that have got abnormal glucose homeostasis? So you can do that through the IMPC portal. You can go in and look for a particular phenotype, abnormal glucose homeostasis. You get summary stats across the whole of IMPC of how many genes have got abnormal glucose homeostasis. But you can go and actually just get the list of all the genes that have got problems with glucose homeostasis, from COL4A5 through CPE and so on and so forth. And of course, you can drill down on any gene and look at the individual data, so you can assure yourself that the data is robust and meaningful. You can download the raw data if you require as well. The other thing that you can do, of course, is to ask, does my gene model a human disease? Or for a particular human disease, is there an IMPC mouse model? And Damien Smedley at the Sanger has developed this tool called Phenotype, which uses, I won't go into the details of it, uses semantic similarities between how we describe phenotypes in the mouse and human to be able to identify potential models in IMPC for human diseases. So there's now a tool in IMPC where, as I said, you can actually look at a particular human disease and ask whether there is a model which seems to replicate those features of human disease, or if you've got a mouse with particular phenotypes, does it seem to model a particular human disease? And this is obviously tremendously powerful in terms of the utility of mouse mutants. Here's an example here. Hermansky-Pudlak syndrome 7, and asking the question, are there potential mouse models where in fact there are some? You get a score, a phenotype score. The closer to 100 that score is, the better the model. And indeed, you can drill down on those scores to actually look at the common features between the human and mouse diseases. So here are the major features that are seen in the human disease, the Hermansky-Pudlak syndrome, and those are the features that are seen in the different mouse models that are in common with the human disease. So you can make some assessment of how good you think your mouse model is for human disease. So do go to the site. You know, Hugh took you through it yesterday. It's a fantastic site for just playing around and finding out new information about gene function and its relationship to disease. And this site will continue to grow because by this time next year, we expect to have 3,000 or 4,000 genes phenotyped. And of course, over the next 5 years, we intend to grow that to try and cover the whole genome. But let me just show you a few examples of random genes. Every week, well, not every week, every month in the IMPC, we have a big conference call where all the centers come together. And they could do this every week. They could do it practically every day now. A new gene, it's never been looked at before. What are the phenotypes? So we're constantly showing these examples where we've uncovered new information that's just never been there before. And they're often for genes that we know virtually nothing about. I mean, here's a gene here, neighbor of BRCA1 gene 1. Well, it just happens to be close to the BRCA1 gene. Nobody knows what this gene does. It's never been looked at before. A mutant's never been made before. But you can see it's got some very clear phenotypes. Here it has some interesting expression patterns in the brain, in other organs, in the esophagus, and so on. And but it also has abnormal glucose homeostasis, very clearly here, but expression in a broad range of organs. And maybe that abnormal glucose homeostasis is potentially related to some hypothalamic expression or something like that. But this just shows you the power of looking at mice from this perspective. It's an enormously powerful tool for generating new hypotheses about genes in the genome and starting new lines of inquiry. So there's one gene. Here's another gene. Never been looked at before. Nobody really knows much about it. Death-associated protein kinase 1. Has a very interesting retinal phenotype that was picked up in the primary phenotyping platform, which has been confirmed now with histology. You can see this clearly disformed retinal layer here. So again, completely novel phenotype for a gene that really hasn't been explored before. But it's having that information at your fingertips as you do your mouse genetics that's going to be absolutely clear. So I wanted to come back to LMOD1 because we phenotyped LMOD1 in Umodic. We also phenotyped it again in IMPC. We get the same data, so that's good. But in fact, you might be quite interested. This is quite a good paper. I think it came out in 2013. It's from Robert Williams' lab in PLOS1. Functionally enigmatic genes, a case study of the brain ignorome. And what Robin Williams was very interested to do was to look at all of the genes that are expressed in the brain, and there's a very large number for which there is no information whatsoever. And he classified ignorome genes as genes which had either 0 or 1 PubMed hit. So virtually nothing is known about it. And LMOD1 falls into that class of genes. In fact, it's mentioned in this paper. It says, here's a gene. It's known to be expressed in the brain, but we know nothing about it. So LMOD gene belongs to the large class of genes expressed in the brain for which there's no functional information whatsoever. Now we have that information through IMPC. We're able to go in and look at all the various phenotypes, including neurological phenotypes that might be related to its expression in brain. So just to come back to this heat map for LMOD1 on IMPC, you can see that there are nervous system phenotypes. There are sensory phenotypes, there are behavior and neurological phenotypes. There are phenotypes that correlate with the places in which it's expressed. So this provides the start point for understanding not just what's happening in the brain ignorome, but if you like, the liver ignorome, the pancreatic ignorome, and so on and so forth. Of course, if we look across all of the phenotyping information in IMPC, and we classify the phenotype annotations according to the different areas in which we've made phenotyping assignments, at a higher level ontological analysis across the different areas, such as mortality, aging, homeostasis, metabolism, behavioral, neurological, skeleton, you can see that in most areas we're getting very significant phenotype hits. Of course, in some areas, the respiratory system's an example, we're not doing so well. We're not getting so many genes where we see phenotypes. And that partly reflects, of course, the phenotyping pipeline we have. It isn't perfect. We always want to improve it. We'd like to get more information for the same amount of effort. So this kind of graph indicates that we're doing pretty well in getting broad brush comprehensive phenotype information, but in some areas we need to do better. The same is applied if we look at the hit rates, the number of annotations we're getting according to the different phenotyping platforms we're using, all the way from Viability primary screen through clinical blood chemistry, body composition, DEXA, and so on, right through down to some tests where we're not getting very many hits. That might partly reflect that there aren't that many genes which will give us hits, but almost certainly in some cases, it indicates that we don't have quite the right test for looking at that system. So I want to finish up by coming back to embryonic screening, because I didn't really say very much at the start, but you'll notice that we test all of the mice for fertility, and we see whether the homozygotes are viable or not. And if they're not viable, then they're starting now to go through an embryonic screen. And I want to tell you a bit more about how this embryonic screen works and the kind of information that we're getting from it. So the first is viability. Let's just look at the numbers for viability. And this summarizes all of the numbers to date from lines that have been tested for viability. And you can see that if you look at this panel here, which is analysis, you can see that pretty much across all of IMPC, we find about a quarter of the lines are lethal. That is, that they don't survive to term. And about 10, 11% are subviable. That is, some animals will come to term, but many will die in utero. So overall, if we include lethal and subviable, this comes out about 35% of the lines need to be looked at embryonically. So I want to tell you in a bit more detail about what the embryonic screen is. Just focusing on that particular area of the IMPC phenotyping pipeline. This summarizes what generally is happening at most of the mouse clinics in analyzing mice that are embryonic lethal, that is, that they don't survive to go into the adult pipeline, and they then go into this embryonic lethal pipeline. And first of all, most of the mice, the embryos are looked at E12.5. We look to see if they're viable at that age, and we look at their gross morphology. And if they're alive at that age, E12.5, we'll then go to later time points, such as E15.5 or E18.5. Again, we'll look at viability at those ages. But importantly, we'll look not only at gross morphology, but we'll use sophisticated imaging techniques, such as micro-CT and HREM, or in some cases MRI, to look at the morphology and the anatomy of those embryos, and to try and work out why they're dying and what's wrong with them. Of course, if the embryos are dead at E12.5, we go earlier. We look at the viability at E9.5, we look at gross morphology, and we also use another imaging technique called optical, name's gone actually, OPT, to actually image embryos at that particular stage, optical projected tomography. So we have I'm not going to go into all the details, but we have a complex pipeline here of looking at the different stages of embryogenesis for embryonic lethal mutants, using sophisticated imaging technology to look at the embryos at particular stages in order to decipher what might be going wrong with embryogenesis. And this embryo pipeline is beginning to really ramp up at different centers across the world, so that many of the embryonic lethal lines are now being analyzed through this pipeline. And obviously that data is going to grow and grow as well. But I should just emphasize again that irrespective of whether a line is homozygous lethal or not, we'll pick up expression of LAC-Z at this stage of E12.5. And having embryonic expression as well as adult expression is very important for not only understanding phenotypes in the embryo, but also understanding adult phenotypes. So that's a kind of rush through IMPC. There's obviously a huge amount that I could tell you about it, but IMPC never stands still. In terms of future developments, we're looking at the new technologies of making mutants. So in fact, in IMPC, we want to make our mutants faster and cheaper, particularly in phase 2. And I should say that that standardized knockout first conditional allele is to some extent on its way out. We'll continue to use it for a while, but we're actually looking at, I'm sure this is a technology you've heard of, I'm happy to discuss it with you further. We're looking at CRISPR-Cas9 technology for now making IMPC alleles in the future. And there are various pilots underway at IMPC centers to inform how we'll use CRISPR-Cas9 in the future. and the kind of allele that we'll make. But importantly, whatever we make, it will be a standardized, high-quality, gold standard allele that we'll make. And of course, future developments in phenotyping. For phase 2, we're looking at introducing some quite complex phenotyping technology, which will give us more data, more sophisticated data at lower cost, and also longitudinal data. For example, home cage monitoring. Monitoring animals in their home cage over time, looking at behavioral and social interactions and so on. There are very sophisticated tools now that we're developing for doing that. And of course, looking at other possibilities for phenotyping, including having an aging component to the IMPC screen, which I talked about earlier. So in conclusion, IMPC is set to deliver 5,000 mice lines and the associated phenotype information by 2016. We're pretty much on target for that in a couple of years' time, by the end of 2016. Phenotype data from the first 1,800 phenotype lines is available, and you were looking at some of that yesterday. Please continue to look at it. We're planning phase 2 to finish the genome, and we do believe that that catalog of mammalian gene function that we're generating will be transformative for biology and biomedical sciences and will be real utility to everybody who's doing mouse genetics around the world. And we've also got a Twitter account as well. So if you look on Twitter, you'll see tweets of new phenotypes, etc., that have been developed. So that's kind of fun as well. These are all the groups around the world who are involved in IMPC. I couldn't possibly put up a slide with everybody's name on it. It would cover slides and slides. But there are hundreds of people involved, but these are all the major centers involved. But I think it is worth, partly because I talked a lot about it, and you heard a lot from Hugh yesterday about it, it is worth acknowledging the Informatics group for MPI2, who've done such a major job at the Data Coordination Center, the IMITS, the tracking of all the work that's going on, and the Core Data Archive at EBI as well. So thank you to all those people.